Well, thank you for that very nice introduction. It's always been great having conversations with you over the years. Makes the work better. And more broadly, thanks to the organizers for this meeting. It's a sort of a nice retreat from the noisy world around us and getting a chance to spend a few days talking science with friends and colleagues. There's really nothing better right now. So with that, the title of the talk is The Neural Basis of Flexible Auditory Learning. But before getting to sort of our bread and butter, I just want to talk a little bit about babies first, right? And so many of us have experience with babies, right? It doesn't seem like they do very much. They drink their milk, they sleep a lot, and unfortunately we have to change a lot of diapers, right? And so William James referred to this as the blooming, buzzing confusion of that early infancy period. And so here are some pictures of my daughter before she could walk, before she could talk, but you could see that there was something going on in the way she looked at the world. And so some really clever developmental cognitive scientists operationalized that to be able to demonstrate that babies will look at things longer that violate their expectations than things that don't, right? And in doing so, recognize that infants have a very rich internal mental experience. It's not just that they're peeing, pooping, and sleeping, but they actually have an intuitive understanding of arithmetic operations, logic, and can even do some basic forms of hypothesis testing, right? And so this suggests, right, that what you know can be very different than what you show and fundamentally depends on the behavioral report that you use to measure it. And so this has been demonstrated in babies, in non-human primates, and even to some extent in birds. And our thought is if we could do this in rodents, it unlocks the opportunity to understand the neural basis of this. And so how do we do that? Well, we kind of reduce it down to a simple goal-directed auditory go-no-go task where mice are trained to lick in response to a target stimulus and get a water reward and withhold licking to a foil or S- stimulus. And here's what the behavior looks like. These are the task that we use uses pure tones that are 3/4 octave spaced. So this is a pretty easy task perceptually for the mouse. And what we're really trying to understand are some associative computations, which I'll get into in a bit. And here's what the behavior looks like, if it works. Go. The animal here's a target stimulus. This is a head-fixed mouse. Licks are detected with an optical beam, and water comes out of this lick tube. You can see the animal is expert, licking correctly to the target, and also withholding licking to the foil stimulus. Okay, pretty simple task. And so what we observe as animals learn this when we look in the reinforced context, right? So this is when animals have a lick tube present. Here's what the behavior looks like at a much earlier time point in learning. We'll see that the animal is sitting there. Hears a target sound, licks and gets his water reward, but is also incorrectly licking to the foil tone. Okay, it's patiently waiting for the next stimulus, meaning it's not licking all the time, but it's going to lick in response to the target to get its water reward, but also unfortunately lick to the foil tone. And so we would characterize this mouse as having learned something. It's responding to stimuli, but it's not yet discriminating. And we can see that when we quantify the performance of this animal. It's performing in near chance levels. So then we did this small trick where we remove access to the reinforcer by pulling out the lick tube, okay? And here's what the behavior looks like. Same mouse, same day, same session. And what we see is that, funny enough, the animal licks to the target stimulus in the air and can do so reliably for a small number of trials, but when it hears the foil tone, the animal doesn't lick at all. It's licking aggressively in response to the target stimulus, but even in this slightly agitated state, you can tell from its nose moving, it doesn't lick at all to the foil. And so what we concluded from that when we calculate the behavior is that this animal's performing at expert levels, right? Same day, same session, same mouse. And now if we plot this over learning, what we find is rather than the slow, negatively accelerated learning curve that many of us are used to, we see a much more rapid, one could argue, insight-like acquisition of the task contingencies. And so we can think about this black line not as a measure of the juridical learning, but actually what the animal is choosing to express or is expressing on those trials, versus the gray line here, which is likely closer to the core acquisition of the task contingencies. And so what this suggested to us is that reinforcement is required for learning, but can paradoxically mask the underlying task knowledge. And so we were able to demonstrate that with Eve in ferrets, and also with Peter Holland using old data and freely moving rats in a wide variety of task designs. And so this raises the question of, well, what is the role of the auditory cortex then? And so when we think about the history, there's a long history, as we know in this room, about the role of the sensory cortex broadly, but auditory cortex specifically in this type of learning. And we can think about learning with a very simple heuristic, an SRO framework, in which there's bidirectional relationships between stimulus and outcome, stimulus and response and outcome. And the way we tend to think about what the sensory cortex is doing is somehow related to increasing the salience of that stimulus signal through an increase in stimulus selectivity, expansion of cortical representation. And we tend to think of those, and people have shown that these are sort of somewhat correlated with the performance of the animal. But what I've just showed you, though, is that learning and performance are not one and the same, right? And so we wanted to understand, well, what is the role of the auditory cortex in this type of discriminative instrumental learning? And so the first thing we needed to do, and Jean-Marc alluded to this yesterday, is to determine whether or not the auditory cortex is even involved in this type of task, right? And so what we wanted to do, though, was try to figure out what is its particular role during learning. And so what we can use is optogenetics here, where we can inactivate the auditory cortex on probabilistically at 90% of trials when the lick tube is in. And we do that by shining blue light in animals that express channel rhodopsin and parvalbumin positive neurons. It's a very efficient way to silence the cortex. And then instead of measuring performance on these trials, we actually measure performance on our probe trials when the lick tube is out and the light is off and the auditory cortex is back online. And when we do that in our control mice, we see the animals rapidly acquire the task contingencies, whereas in our test mice, what we identify is a significant degrading of that learning process, suggesting that the auditory cortex is used during learning. And then what we can do is take advantage of our design and compare performance when the auditory cortex is silent versus when the auditory cortex is active with the lick tube in, right? So this is sort of a apples to apples comparison of performance during learning. And what we find in control mice when we plot accuracy, when we compare light on versus light off, is a flat line, okay? Because there's no optogenetics, there's no channel rhodopsin, and we see that there is no effect of light. Now, when we do that in our channel rhodopsin mice, however, what we find is that there's a significant effect of silencing the auditory cortex early and intermediate periods of learning, but by the end of learning, it seems to go away, right? And so this suggested to us that perhaps the auditory cortex is involved during learning, but not at expert. But just to rule out the possibility that it's because the animal's sort of getting used to the light, right, over time, and then it's sort of improving, we took a separate group of animals and just silenced at expert levels, and we see no difference between light on and light off, which is very consistent with work from Brice's group and others, suggesting that the auditory cortex in this type of task is not used for expert level decision making, suggesting it has a particular role during learning. And so we conclude here that the auditory cortex is the default pathway for this type of discriminative learning. And then we wanted to ask, well, what's actually happening, right? What's the nature and form of plasticity that may underlie this? And so we classically think of what the auditory cortex is doing is related to sort of perceptual, as I mentioned. But one question we really wanted to pursue was whether or not there's any evidence of associative computations that are happening in real time. To do this, we use large-scale two-photon calcium imaging, which multiple people have talked about. We target layer 2-3. We can image largely excitatory neurons in this area. And we can do this both in animals that learn the task, right? So these are animals that over the period of time we image over the entire course of learning, and the animals are learning. And we have a separate group of animals that are passively exposed to the same number of tones over the same number of days, but never learn the task itself. And this serves as a base case comparison to understand what the auditory cortex is doing. And I'll refer to this as learning mice or learning networks or passive mice and passive networks. Now, one of the things that we wanted to do was to be able to track the exact same neurons over the entire course of learning. And here we have 6 example neurons where we've tracked for every single day that the animal has heard these stimuli and performed the task. Like this, we've tracked about 2,500 neurons in learning animals and a similar number in our passive cohort as well. Now, one fundamental question we wanted to ask is in this type of discriminative learning, is there any evidence of map expansion, right? Because we have the topography, right, from our two-photon imaging. And so we can look at the tuning of individual neurons before the animal has learned. So this is in what we call a tuning curve session, purely passive, right? And then we know where our S plus and our S minus is, and we can calculate the representational space here. Now, the expected scenario we thought would be that we could see an increase in the representation of the S plus over learning, Or given that this task has a strong behavioral requirement to inhibit licking to the S-, perhaps we would see an increase in both. Now, what we actually saw, however, was not that, right? We did not see any increase in the representation of S+ or S-, and if anything, we saw a slight decrease, suggesting that map expansion does not appear to be, in this task, the core substrate of the learning process. So then what is it? So we kind of went back to the drawing board and said, well, this is a massive data set, right? Here's one neuron responding to a tone in one trial. We have thousands of trials over learning. We have our different trial types: hits, misses, correct rejects, and false alarms. And of course, we have all our thousands of neurons. So how do we make sense of this? Well, we chose to use an unsupervised low-rank tensor decomposition approach to allow us to reveal the underlying neural dynamics that explain variability across multiple timescales. And so, what we were able to identify with this, and this is just a t-SNE visualization of the neuronal weights that come out of the tensor decomposition, are 6 largely independent, but with some overlap, dynamics that seem to drive most of the learning-related processes. Now, we see that 2 of these dynamics relate to the passive network, and 4 of them relate to the learning network. We can actually map dynamics onto individual neurons. It's a bit of a complicated approach, but happy to talk about how we do that. And what we saw in this is that 4 of these groups of neurons seem to be related to classical things that we would expect the auditory cortex to do, which is a stimulus-evoked response. But then 2 of them seem to be very different. They did not have your classical stimulus-evoked response, either at onset or offset. Instead, they had activity that we kind of initially termed as latent trial. And for a few minutes, I'd like to focus on one particular ensemble of neurons that we think is performing a core computation that relates more to a higher order function rather than something related to specifically auditory processing. And so in this group of neurons, which is present in every learning animal, we can take a look and see what its activity looks like. So here we're going to look at hit trials, where the animal hears the S+, licks correctly, and gets a water reward. On the y-axis, we have hit trials in 20 trial blocks, okay? This line here indicates when the animal has acquired the task contingencies, meaning it's performing really well in the probe trials, and this line is expression. So this essentially is all across the entire course of learning, and then the x-axis here is time in trial. And so what we see in the first 20 trial block is a nice stimulus-evoked response, but then as we kind of move up and as the animal continues to learn, we see that this seems to be changing. And instead of a stimulus-evoked response, this group of neurons is encoding something much different later in the trial that gets stronger and stronger over time. And then when the animal is expert, it starts to fade, right? And this got us very interested that perhaps this is some sort of higher order computation. And so we can zoom in on the first few trials, and we can see that this latent trial signal is emerging extremely rapidly. So rather than taking days to happen, we're seeing it within 20 to 40 trials emerge. And so we wanted to ask, well, is this some sort of methodological problem? Calcium imaging is great for many reasons, but also has lots of challenges. And so we could actually look at another trial type, which is the S-, the same group of neurons. We're not seeing this slow activity. We see a very precise, beautiful stimulus-evoked response. So we don't think this is because there's something wrong with our calcium reporter. Another possibility is that this could be a slow sensory response, something that's learned. And so now we can look at trial-by-trial data in an example mouse where we look at hit trials where the animal is licking, that's what these black circles mean, versus missed trials where the animal does not execute the instrumental lick. And we see that on those missed trials, there's no such latent trial activity. We see them on hit trials, but not missed trials, so on and so forth. And so when we compute this across the entire population, we see a significant activity on the hit trials, latent trial, but not misses. Now, many of you are probably thinking that, well, these black dots seem to be related to that, so perhaps this is a lick-related signal. And of course, that's something we were very interested in, to try to understand the nature of this latent trial signal. And so the first thing we wanted to ask is this related to the lick bouts, right? When an animal executes a lick bout, perhaps that signal is getting into the auditory cortex and reflected in the neural activity. And to look at this, we looked at lick bouts outside of the task context, either the intertrial interval, or when we first put the animal into the apparatus. This is a lick histogram. This is the activity of those neurons, and we see no such latent trial or ramping activity, no correlation between licks and this particular group of neurons. So it's not that. Then another possibility, right, is that it's task-related lick initiation, right, which is that, the animal's executing a lick, but only within the task context. And to understand this, we looked at our false alarm trials late in learning, right? So these are trials where the animal incorrectly licks but does not receive a water reward, and we see no such activity there. So we don't think that it's task-related lick initiation. Then the final thing that we could test was whether or not it was reward consumption. And remember, we have two really nice trial types: those in which the lick tube is present and the animal is rewarded, and those in which the lick tube is not present and the animal is not rewarded. And so we can look at those, and so we have very different lick structures between reinforced and probe, and no water on probe trials, but we still see a big latent trial signal. And altogether, this allowed us to conclude that what we're actually observing is a reward prediction signal. That this signal is related to the animal's internal expectation of receiving reward at a particular time. And it's not related to consumption or action. Now, I think we have heard, at least in the mouse literature these days, that movement, reward, it's everywhere. And so to what extent is this actually true? And to what extent is this actually important for learning of the task? And to do this, what we did was a closed-loop optogenetic approach, where we can, on S+ trials, detect a lick, and then turn on our optogenetic silencing, and silence during that reward period, okay? And so this is allowing us to impact the learning process, but not performance on the current trial. And what we find is quite striking, where we see a delay in both the learning process, but also an increase in the response latency, suggesting that this reward period activity, this reward prediction activity, is actually critical for the learning process itself. We see this also when we do this closed-loop silencing on false alarm trials, suggesting that the reward prediction signal is truly a teaching signal in this network. Now, the other thing that sort of got us very interested is, on some level, this is now neural activity that we can look at in real time, and could we infer the nature or driver of errors, right, when the animal is doing the task? And the particular error that happens often is a false alarm, right, the animal licking incorrectly to the S-. And so during expression, as I mentioned, right, when the animal has already learned the contingencies but continues to exhibit some errors, we see that false alarm trials do not have a latent trial signal. Right? So there's no reward prediction signal, even though the animal has executed a lick. And so we think of these as RP- errors, and that they're largely exploratory errors. I won't sort of defend that except to kind of point to a paper by a graduate student in the lab, really demonstrating that the vast majority of errors during this period are driven by a form of structured exploration. But what's happening during acquisition on these probe trials, right? The animal still sometimes makes some errors at that early time period. And when we look at those false alarms, we see something very different. We see a significant latent trial bump in activity, okay? Suggesting that on these trials, when it's early in learning, the animal hears the S-, licks, and is in fact expecting a reward, right? Suggesting that these are RP+ errors, and one possible interpretation is that it's a true knowledge error. The animal does not know that it will not be rewarded, right? And so we said, well, could we actually classify errors based on these two sort of neural phenotypes, right? No reward prediction signal and with a reward prediction signal. And so we can then look in the reinforced, now the lick tube is present, to understand, well, when is an animal making a knowledge-based error versus something that may be more exploratory? And so if we look at individual trials, we can see that there's a sequence of trials. Now this is time in trial, so this is the stimulus here. And what we see on these three trials is a latent trial activity, a reward prediction signal. But not every false alarm trial is that. What we find, and we can classify these trials based on whether the latent trial signal is there or not, is that we see sequences of reward prediction error trials and some without, suggesting that not all errors are equal. When we actually look at the lick activity, there's nothing in the lick activity that helps us figure that out. It's really by looking at the neural activity that we can, in a way, read the mind of the mouse. Why is it making such an error? Now, we can then ask, well, to what extent is the pre-learning tuning of these neurons predictive of whether it will become a reward prediction neuron? I didn't mention this other group, this other computation that we observe in Ensemble 6, which is related to the other stimulus and action suppression. But what we can do is we can look at these neurons, and then we can look at their pre-stimulus, pre-learning selectivity index. And what we find is that there's no correlation between neurons that have a preference for the S plus and becoming reward prediction neurons, or preference for the S minus and becoming action suppression neurons. And that is just quantified here. And so overall, we think that this suggests that the auditory cortex is critically involved in the early acquisition of this task, but then essentially maybe training itself out. And that the role that it plays is in enabling a higher order computation of reward prediction. What I didn't show is that this seems to happen in a spatially segregated population as separate from an action suppression signal that's more related to the performance improvements over time. Now, interestingly, these are completely uncoupled to the stimulus properties, stimulus response, whether it be individual neurons or tonotopy. Now, what we're interested in the lab is sort of not just understanding this behavior in the excitatory network, but what is actually the broader circuit that's involved in this. And so, in particular, in the local microcircuit, we're both interested in inhibitory interneurons, and also a postdoc in the lab, Dr. Charlene Moore, brought her fascination with glial cells to try to explore the role of astrocytes in this process. And so what she's done is being able to get GCamp into astrocytes, longitudinally develop new tools to be able to look at both the cell bodies, but also potentially processes. And in doing so, and some slight changes to the task structure, because calcium signals in astrocytes are much slower, was able to demonstrate so far, and she had a poster yesterday, but she's around, so you should definitely find her and talk to her, that the astrocytes develop what appears to be a pure sensory response to the S plus within just the first 100 trials. That's a lot more interesting things that are happening here, so feel free to talk to her. And importantly, Charlene is on the faculty job market, and this is just one of 3 projects that she's had in the lab. She's been very interested in looking at auditory processing in different contexts, both habits and Alzheimer's disease. She's a fantastic scientist colleague, and I urge you to talk to her and find her. She's right here in the middle here. Another way we're following this up is the role of neuromodulation. And Dr. Jenny Lawler is very interested in the role of ascending cholinergic neuromodulation from the basal forebrain. And so similarly, in some ways, one of the biggest challenges is understanding to what extent is this neuromodulatory signal homogeneous or heterogeneous, right? And so what she can use is 2-color, 2-photon imaging to look at cholinergic axons here in green and cortical neurons in red, and try to use computational approaches to segment these axons and look at them all across learning. And so this is something that she's doing now, and also using optogenetics. Check out her poster on Thursday, P58. And also Jenny is on the faculty job market as well, and has done some really amazing work. She joined the lab having worked on 2 species in humans and ferrets, and decided to do the same thing as a postdoc, but this time in mice and in echolocating bats, and really has done some beautiful work looking at categorical primitives in the inferior colliculus of the echolocating bat. Now, as I mentioned, one of the really interesting things that I think we discovered was that the auditory cortex appears to kind of train itself out of the task, right? And so what is happening in this expert period? What part of the auditory system is, in a way, the sort of final common pathway to perform this task? And Sue, a graduate student here, she's also here, is working on this. And she has some really interesting data using temporally precise optogenetics bilaterally in the medial geniculate body to try to understand the role of the MGB. This is part of a broader question around what is the nature of this tutoring between cortical and subcortical structures. Her poster is on Thursday, P31. Now, in the last couple of minutes, what I'd like to do is kind of talk about where the lab is going. Now, head-fixed behaviors are great for some reasons, but Jenny Bisley pointed out some of the challenges that they have. And one of them that's relevant for us is that we're really interested in learning, but learning across the lifetime. How is it that humans and other animals are able to learn not just one thing, 2 things, but potentially 10, 20, and flexibly recombine knowledge to perform new tasks? Now, to address this, a very talented graduate student, Anish Ball, in concert with postdoc Andrea Santi, decided, well, we got to move away from sort of standard training approaches and really think about this in a home cage design. And so what they built is essentially a home cage training environment in which animals live in a center home cage and then can run through tubes to perform tasks in various arenas. And so what we find is that the animals seem to like this. I don't know how to interpret that, but at least they seem to, right? They run readily through these tubes, perform the tasks, and they have all their food here, and the only place they can get water is in these behavioral arenas. And so one of the thoughts is, well, how do we actually look at questions around continual learning? And so we needed to parametrize the space in some way. And so one simple way that we're thinking about is that we can kind of think of one axis as the structure of the task, right? Whether that be a go, no-go task or a two alternative force choice, delayed match to sample, so on and so forth. Another axis would be the perceptual dimension. And within audition, you have a number of different perceptual dimensions, frequency, duration, so on and so forth, right? You can also think about a rule dimension, which let's imagine like a reversal. And the intersection of this, one could consider, is the definition of a task in this framework, okay? Now, this matrix approach allows us to start asking what I hope are interesting questions. And so in a preliminary set of studies, we developed an 8-task learning sequence in which mice learn 4 sequential go-no-go tasks, okay? Then they switch the report to be a 2 alternative force choice, and they learn 2 more 2 alternative force choices. These are all on different perceptual dimensions. Then what we can do is test them on tasks where they've seen both problems before, a 2 alternative force choice and a frequency discrimination, but never together, okay? And ask, well, what do they do? So in this particular design, it's not the 4-arena design I showed you before, but this is where animals are living in this home cage here, and they go into one arena, right? They'll run into a particular arena. In this arena, and here's this mouse performing a 2 alternative force choice. It hears a sound, nose pokes left, and goes up for its water reward. And then it has I think there's a go right trial here as well, okay? Now, one of the really great things that I think we were able to see is that mice seem to learn in this environment extremely well. So here are our 8 tasks over time, our go, no-goes, and our 2 alternative force choice. We're plotting accuracy per task in the colors. And what we find is that mice rapidly learn these tasks. They can learn this 8-task sequence in about 40 days, and we can also do retention tests, and we can demonstrate that the animals have retained the task contingencies. But as we dug into individual animals, we started to see some interesting stuff. So remember, we have these 6 tasks in sequence in which there's something new to be learned, and not surprisingly, this particular animal starts at chance in these tasks, okay? Then we can test for inference or compositionality on these 2 tasks. And what we see in this mouse is that immediately when starting this task, the animal's performing at expert levels, both in task 7 and really very much so in task 8, okay? And suggesting that the animal's doing definitely something more interesting than just a rote relearning. And so here's an example mouse where we see that effect in task 8, but then not all mice do this. We have another mouse that starts at chance. We can look at the individual behavioral actions, and we see that in this mouse, we see that the animal is really performing poorly at the beginning, whereas this mouse, immediately from the beginning, is doing very well. And these mice are volitionally entering the arena and initiating a trial. But what we find in the first few trials are some misses. That's what the gray bars are. Now, our fanciful idea is that's when all the magic is happening. The animal's hearing the sound, it's recognizing something, and so something is happening in these first few trials where it's doing that calculation, well, let me just apply what I know to this new task design. And so we see that about half our mice show this type of compositional behavior by task 8, and half of them don't. We don't believe that these mice never will, right? There's still additional tests. Perhaps they just need more evidence before they apply these higher order rules. And so we've gone to scale. We now have a full rack training about 70 to 80 mice at a time. We're looking at this in sort of adult animals, but also aging. And we're trying to integrate some tools to be able to record the brain and also working with some theorists to try to model that. And so overall, we're sort of interested in this brain scale model of continual lifelong learning, looking at local circuits in the auditory cortex, ascending neuromodulation, cortico-subcortical tuning, and how these signals get turned to action. And sort of in a big sort of future direction is looking at continual multitask learning. And so with that, I'd like to thank you. I'm happy to answer any questions, and also all the members of the lab and our funding sources. Thanks.
Thank you, Kishore, for a very exciting talk. If you have questions, I would like you to raise your hand, and we'll pass this around. I want to begin by one question. Why does it take so much longer for suppression than prediction?
Yeah, I mean, so we think of it as like a performance optimism. So in a go, no-go task, where there's no cost to licking, that the animal has just essentially made the decision that it's okay with extra time. The cost is time, so reward rate goes down. And so we think that's the biggest driver for why there's so much variability, right? Some animals, they really want to optimize that quickly, other animals don't. Why? I don't know.
Okay.
Recording the ACC maybe, or the Insula? Yeah.
All right. Thank you.
Okay.
One more question here.
Hi,
Oh, yeah.
Maria Geffen here from University of Pennsylvania. Kishore, this was really beautiful. I'm really fascinated by your analysis with the online feedback where you can actually modulate the learning curve. I'm curious does that analysis select for the cell ensemble that's reward predicting?
No.
And what does that mean? Because presumably you're also activating your, what did you call them, like the error network or the,
Oh, yeah.
Sorry, the
we're
other
activating every
popu-
neuron, most like, or
Well,
suppressing all activity.
suppressing,
Yeah.
so you're also suppressing, what's the other ensemble?
The
Sorry.
action
The
suppression.
action
Yep.
suppression.
Yeah.
What does that like mean? And also, have you given thought to doing this like in real online
Yeah,
kind of
absolutely.
like
So,
identity?
it would be wonderful to be able to suppress specifically the reward prediction neurons. For that, we'd need to use something like an SLM.
Yeah.
The problem with that would also probably be that we would be able to do for a small number of neurons, but not the sort of
Right.
whole network. But I think if we did that, the learning effects would be even stronger. But
And-
here, because we're suppressing activity, we're not driving additional activity. We think that if it's serving as a teaching signal, that's sufficient to get the behavioral effects.
And how do you identify those ensembles?
Yeah, so, well, in the optogenetics, those are not done during imaging, so we don't identify them. But the way we identified them was using the tensor decomposition.
Okay.
And so we can they get classified sort of naturally through that. But we can also use hierarchical clustering, and we sort of identify the same group of neurons. And we've also run an encoding GLM, and we can see that these reward prediction neurons sort of pop out in all of those analytical approaches.
So with tensor analysis, you're running that against all the variables that include the reward and the...
Yeah, so the way we do the tensor, so we actually put all of our animals together. We take our learning animals, we align them based on their behavioral metrics. We also put our learning networks, passive networks, meaning animals that don't learn the task but hear the tones. And then we run the decomposition on that full, massive, what we like to call a mega mouse, to identify shared and distinct variability classes. And there's really nothing for the tensor decomposition that you're doing other than letting it sort of identify the sources of variability.
Yeah, thanks. Yeah, that was a really great talk. Since you, yeah. So you can see me.
Yeah.
But okay, I'll stand. Fine. So you can hear me now. So for when you said reward prediction, one thing that comes to mind is I was waiting for you to say error.
Yep.
And I guess, do you see any of them? And also, how coherent are those neurons? Like, there's a small population, but do they all fire together? And does it sort of predict how quickly the animals are learning, like how often they fire?
Yeah, lots of interesting questions. We were very interested in the possibility that we would see error because we have our probe trials, which is where there's no reward. And so we do not have any sort of robust error signal. So we really think of this as the reward prediction signal that then gets likely fed down into the striatum, which is then used to compute error, right? And so we think it's a sort of antecedent to the error signal. One. Two, we see that the population in each mouse is pretty coherent, both in terms of their temporal kinetics, but also in their spatial. They tend to be clustered together. And so why would that be the case? The temporal kinetics, okay, that makes sense. Why would they be clustered together? There could be something just very simple about a neuron that sort of, may not be predetermined to be a reward prediction neuron, but if it becomes one, is more connected to its local partners, and that's computationally efficient. I don't know if that's what you asked, but I think that was
great.
Okay, yeah.
And also, just I guess real quick, does the frequency with which a mouse's like reward predictions fire, you said there's only happening on some trials, does that predict how quickly they learn?
Oh, yeah, we have not done that specific analysis because, with only 5 animals in which we have that data, it's hard to kind of have confidence in that type of thing. But I think it's a great question, yeah.
All right. In the interest of